Good afternoon. Welcome to the presentation on my thesis, which is on the large-scale reanalysis of public proteomics data. And first, I would like to talk a bit about how this data looks like and how I am able to use it. Then I would like to discuss some issues that I encountered along the way, more in particular about the metadata and the complexity of the data. And then I want to show a use case of how this data was used and how it can be used in the future. And I want to close off with some future perspectives on my topic. So typically a mass spectrometer delivers spectra and metadata, which we can compare against the sequence database in order to unravel what was in the original sample. And to do that, we use search engines, and in my case, I use SearchGUI that allows me to use multiple search algorithms simultaneously. And then this is fed into PeptideShaker. It's a tool that does the post-processing, and it collates these results, and it delivers a list of identifications with an FDR value. So these metadata, spectra, and identifications can be stored in public repositories, and my work consists of reusing that, so extracting the data back from PRIDE, and then analyzing it in the same fashion as it originally was done, with the main difference being updated parameters and novel sequences that are in the search space or in the sequence database now. And doing that, we can find, or hopefully find, novel translation products and things that were not considered prior to the analysis. So the first problem I encountered was having issues with the metadata, and to handle that we have a tool called PrideOSAP that was developed at the lab by Niels Hustad initially. And what PrideOSAP does is it looks at the experimental or the analytical metadata, and it tries to reconstruct it so we can convert that into search parameters. And during my thesis, I updated that, so it's more data-driven, and it literally looks at the reported identifications to infer search parameters that are directly usable by SearchGUI, so it's easy to make a pipeline out of it. The second problem is, well, it's not really a problem because it's a good thing, is that PRIDE is continuously growing and the experiments are becoming more diverse, it's more complex, and it's just exploding in volume. And we need something to handle that. And to that end, we developed Platypus. And Platypus is a user-friendly tool. It's a platform or a grid engine, you might call it. And it allows for modular pipelines to be constructed and run on a network of computers. So it's able to handle a lot of data simultaneously. And one of these modular pipelines is Respin, which consists of PrideOSAP, SearchGUI, and PeptideShaker. And we use Respin in several use cases. And the first use case that we had was the assessment of the annotation of long encoding RNAs, or in other words, finding potentially coding long encoding RNAs or evidence for that. And we did the same analysis again. So we do the Respin, PrideOSAP, SearchGUI, PeptideShaker, but this time we included the Linkipedia database or transcripts from that database. And the evidence that we found coming from those sequences we used to annotate Linkipedia, so that was a big success for my project. But simultaneously, well, as expected, we did not find a lot, but simultaneously a lot of these things were being published, and studies were being published that there are a lot of long encoding RNAs that are actually coding. So one of our ideas was that maybe we are missing out on them because we are using mass spectrometry, so we did an in-depth study on that. And one of these studies entailed reprocessing of all mouse data in PRIDE and linking these results to the reports by Schwanhauser et al., who reports on half-life values. And in this plot, you see the bubble size corresponds with the amount of evidence we have for that protein, and we are covering the entire range of reported half-life values. So it does not seem that the degradation of proteins is an issue. However, we also did a similar analysis on blood plasma and proteins reported by Anderson et al., who reports on protein abundance. And there you can see that at the lower abundance regions, we start missing out on proteins, despite the fact that we are covering a range that's over 6 orders in magnitude. But with all this in place, we had the Platypus framework, we had Respin, it was all working. So we decided to go a little bit broader and have a protein-wide study. So I analyzed all the human data in PRIDE, and I compared that to the studies of the draft proteomes by Wilhelm et al. At Kim et al. And on this Sunburst plot, every spike corresponds with a protein, and the red proteins do not imply that they are negative in any way, just a way to show it. but the data is definitely complementary. I mean, there's things that Pride is not picking up that is present in the other samples, and vice versa. So that showed me that having more data is essentially better, and it's also shown in the protein coverage. The more experiments we include, then the average protein coverage is increased. And we have used Respin in other applications, for example, a protein association study, which also helped me reprocess all human data for Dr. Gupta. We did a study in collaboration with Gerben Menschkaart and the small open reading frames assessment. And we did a similar thing with Squanti, where we validated their sequences on long read sequences in murine data as well. So to close off with some future perspective, I would love to have the PRIDE-ASA parameters being distributed through Pride publicly, so the reprocessing becomes a lot easier. And this is in the mindset of Elixir projects. Simultaneously, I would like to have a cloud-based Respin, because now I have a local grid engine, but it would be nice to have a cloud grid engine that allows us to reprocess public data, which is also in the mindset of Elixir. And maybe most importantly, I would like to have established guidelines for the reprocessing of large repository-wide studies, and to have a uniform validation for that. And when that is in place, we can start thinking about building comprehensive public proteomes with all the data that's in Pride to make make proteome atlases for all species. So currently we have a lot of workers doing all the work, automating it for us. That's all nice, but maybe in the not so distant future we can have a semi-open ecosystem that's able to reseed itself and automatically update the knowledge presented by having novel sequences and novel experiments. So with that, I would like to thank the Compomics group, in particular Niels Helstad for his work on PRIDE-ASA, Surya Gupta for the protein association study, Pieter Jan for the Linkipedia collaboration, my promoter Leonard Martens, my co-promoter Harold Barsnes, and Harold and Mark for their work on SearchQuery and Peptide Shaker. And if you have any questions, feel free to ask them now, and thank you for your attention.